Hello, everyone. I guess it's good afternoon. It's going to be present in the afternoon. Sorry that I can't be present to provide this presentation live, and it has to be recorded. My talk is, and again, thanks for inviting me by your farm, Algeria. My talk is about current COVID pandemic and small overview about vaccine from smallpox to current COVID vaccine. So what's new? We need many hours to debate this. However, I'm going to make it in 20 minutes, I believe. This is what was given to me. So small overview about current COVID pandemic. COVID-19 is an infectious disease caused by severe acute respiratory syndrome, COV-2, which is originated in China, likely transmitted by metazoan. As of December 6, 2021, there has been over 265 million confirmed cases of COVID. This is according to WHO. And among them, over 5,254,000 deaths, which is about 2% of people that contracted the virus. I gave a talk about COVID exactly a year ago, December 2020. And by that time, the number were 1/3 of what we have today. So just to give you just some sense that the infection is still threat. As of last week, we are still having up to 51,000 deaths worldwide. Likely about 80% of people do recover from infection without needing any treatment. However, fatal cases occurs. We know more than 25 million deaths, and majority of the diseased people are elderly due to immunosenescence and individuals with comorbidities such as cardiovascular disease, diabetes, and obesity. So vaccination is a long-lasting immunity in all population is the most effective way of establishing herd immunity and stop viral spread. So that's a bit of the advantage of immunization. It does save life and keeps us healthy. The vaccines are one of the most effective public health intervention in history. It does prevent up to 3 million deaths every year. This is according to WHO. With widespread immunization effort, rates of illness caused by vaccine preventable diseases have significantly declined and others have been completely eradicated. Vaccine science is continuously evolving. Currently, vaccines are available to protect against 26 infectious diseases, with many more are under development. So this is a list of current infectious diseases that are prevented by vaccines. And all of those there are 26, and as I mentioned, more are being under development, such as Ebola and Zika virus. And some of them are completely eradicated, such as smallpox. Worldwide, it has been eradicated in 1967. And before, with poliomyelitis, virus was induced in paralysis in thousands of kids. Thanks to the vaccines, this has been under control, even though from time to time there is some outbreak in some countries, but it's prevented by vaccine. Hopefully, in short time, we'll be able to add SARS-CoV-2 to the list of preventable diseases by the vaccine. So vaccine started with the father of immunology, who is Jenner. This is through his study of smallpox. Smallpox dated back to the Egyptian Empire. It was a devastating viral disease leading to 30% death of infected people. In 1796, Jenner made a connection between milkmaids' immunity to smallpox and exposure to cowpox blister pus. Indeed, he noticed that milkmaids, even though they were having smallpox, they have scars, but they don't get sick, and there was no death among them. So he made a connection with a blister that the cows were getting, and given that they were milking them, so they were exposed to the cow pus, and they developed immunity. So to test his hypothesis, Jenner applied a pus taken from cowpox post-pustule and put it in an incision of a boy's arm, and he managed to prevent smallpox by inducing cross-immunity with the cowpox. The word vaccine refers to vacca, Latin name for cow. It was devised by Jenner to describe the protective effect of cowpox against human smallpox. In 1967, following the World Health campaigns, smallpox was completely eradicated, and it was the only human viral infection ever eliminated from Earth. By irony, Jenner never knew that smallpox was induced by a virus or any pathogen. By that time, we didn't know about viruses. Since Jenner, vaccine did evolve, so however differently. Indeed, Jenner's approach has its limit. Not every human disease has an analog that confers immunity without causing the disease itself. Some animal diseases that do jump to human can be deadly to both the animal species, like COVID-19, that is thought to be transmitted by bats. The solution is to work with the pathogen that caused the disease, but strip their power to trigger the disease while still teaching the human system to recognize and neutralize the pathogen in the event of an infection. Up to date, approved vaccines are developed through two ways in order to trigger immune response and clear the infection. We have killed or attenuated pathogen. Vaccine development takes time, and the vaccine must not only be protective, but also safe. And like other drugs that are delivered to sick patients, like cancer patients, prophylactic vaccines such as COVID-19 are administered to healthy individuals. This is why it requires very high safety margin. Where I did work on cancer vaccine within 2 years of development, they were already in cancer patients, because those patients, they don't have any other alternative. If we don't try on them some drugs or vaccine, they will die. Whereas prophylactic vaccine, person is healthy. She will only take the vaccine if she considers that it's safe. And this is why regulatory approval of vaccine is very, very strict, since it's given to healthy individuals. So a small overview of current vaccines that are used for being under development or already approved for SARS-CoV-2. So we have 6 types of vaccine. We have what I mentioned earlier. This is the classical, these two A and B are the classical vaccine that have been used up to date for all the approved vaccines. So it includes heat inactivated or irradiated the pathogen. So what it's done is the virus, the bacteria is inactivated by heat or radiation, and it's used to as a vaccine. There is also live attenuated vaccine. Sorry, Justin, I wanted to mention that current Chinese vaccine from Sinovac is heat inactivated. So what they do is the SARS-CoV-2 is heat inactivated, and it's used with an adjuvant as a vaccine. Live attenuated vaccine are use also the pathogen itself. However, the pathogen is still live, but it can't divide in the body. So the protein that allow it to divide have been eliminated. We have also the subunit vaccine. This one also is popular. Subunit vaccine is a protein, antigenic protein that are selected from the pathogen, and they are usually this is a required adjuvant. It's injected an adjuvant to deliver, to induce an immune response. And this is the case of Novavax. And there's many other under development up to date, more than 74. This was not updated. I believe this was from 2020. We have viral vectors. So other platform use viral vector because as a human, we have in our bodies some virus that are harmful. They have been used as a carrier of the genetic material of the infective pathogen. For example, both AstraZeneca and Janssen use adenovirus. Adenovirus is found in human and it's harmless. So we use it to deliver the antigenic material of the virus. We have DNA vaccine. So DNA vaccine It's a piece of DNA that codes to a protein that is immunogenic. For example, in terms of SARS-CoV-2, there is companies that are using DNA against the spike protein that is used by the virus to enter the lung cells. And to deliver it, we apply electroporation in order to have the DNA delivered into the cells. And finally, we have the Amarina vaccine. There's over 30% under development. This includes the recently approved Pfizer-BioNTech vaccine and Moderna. So this one uses mRNA that encode to the spike protein. This is one of the newest techno platform. However, it's not that new because over 30 years, scientists have been working on this vaccine. And myself, 10 years ago, I did work with Pfizer for mRNA vaccine for a cancer patient, and it has been used for prostate and breast cancer. So this is just since this is one of the newest platform, mRNA vaccine. mRNA vaccine need to be formulated in what we call lipid nanoparticles. Lipid nanoparticles is droplets of fat that made of different lipids, and each lipid has its function. So if we inject RNA directly to the body, it won't reach the right cells. It will be degraded because we have a lot of RNAs in our system. So for this, we have the mRNA that is encapsulated in these droplets. Their size is between 50 and 100 nanometer. So it contains many, as I mentioned, many lipids, and in each of these lipids has its function. For example, the ionizable lipid is used to complex the mRNA that is negatively charged with the positively charged lipid, and the protonization changed with the pH. We have helper lipid that helps with the stability of the nanoparticle, and also to fuse with membranes in the cells. Of course, there is also cholesterol, again, to trigger stability, and also PEG lipid that confers protection to the nanoparticles. This way, it's protected against aggregation and also quick clearance. Yeah, the PEG is used to stabilize and protect the nanoparticles from fast clearance. So how it works, the mRNA, when it's injected intramuscular, it's taken by muscular cells, muscle cells. So the nanoparticles are endocytosed by the cells, and it goes into endosome, and in the endosome, given the formulations, the physiogenic lipids with the pH, acidic pH of the liposome, of the endosome, sorry, they diffuse and they release the nucleic acid material, and here mRNA, into the cytoplasm. And the mRNA will use the cell machinery, including ribosome, to translate the mRNA into protein. The protein is excreted and taken by antigen-presenting cells to the lymphatic system to trigger specific immune response, including humoral response that release antibodies, and cellular response that kills directly infected cells. So this is how a vaccine works. So in ideal world, a good vaccine will work like a virus. So people who recover from an infection, this is what we're having, and this is what we're looking in the vaccine. So when a virus is infect a person, the initial defense is what we call innate immune response. So innate immune response is triggered by the virus, because the virus contains what we call pathogen-associated molecular pattern, that it's a signature that alarm our innate immune system that we have an intrusion, foreign intrusion. So this is the first defense, the innate immune response, that is triggered by dendritic cells activation and uptake of the viral antigen. Dendritic cell, this is what we call antigen-presenting cells. So they will take the virus, pieces of the virus, and present it at their surface to induce immune response. So from after the innate immune response, we have adaptive immune response, which is specific to the virus. And this includes cytokine production, Treg regulation of inflammation, because you don't want too much inflammation because it can trigger cytokine storm. You just want the right inflammation to clear the virus and produce lasting immunity. We have CTL, what we call cytotoxic T cell killing. It kills infected cells. It recognizes cells that has a virus inside and it will kill it specifically. And finally, we have T follicular helper, which induce antibody production by B cells. So after an adaptive immune response is produced and the virus is cleared, we still have, even the virus is cleared, we still have immune memory response to SARS-CoV-2. And immune memory, this is what all we are looking for in a good vaccine, specifically for recurrent infection, as long as the virus is not eradicated like smallpox, we still have SARS. But in order to be able, for a person to be able to clear the virus again, if it encounters it second time, we need this memory response. Memory response is a small pool of specific T cells and B cells that produce antibody that are stored in our body. Like that, in the future, if we encounter the virus again, this immune memory T cells will start to divide and clear the virus. So in terms of kinetics, so the innate immune response lasts days to weeks since initiation to clearance. Then we have the adaptive immune response that can be initiated between 2 weeks up to a few months. For example, in terms of SARS-CoV-2 Amarina Vaccine, within 2 weeks, we have B-cell immune response. And then to build a human memory response, we need months and sometimes years. So depending on the vaccine, some vaccines are quite strong and the pool immune response lasts longer. And usually we don't need a boost. However, some other vaccine might require more frequent boosts. So this is a classical step for vaccine development. So usually to produce a vaccine, it takes 10 to 15 years. So initially there is a proof of concept, clinical work in animals and in vitro. Would take 2 to 3 years, followed by phase 1 in order to assess safety in few patients. Then we have phase 2, which also look at safety and also some efficacy in a higher number of patients. And finally, a phase 3 that involves thousands, especially for prophylactic vaccine, it involves thousands of injection to prove efficacy as well as safety. And then after phase 3, there's submission to regulatory approval. And once it's approved, only once it's approved, that the manufacture of the vaccine is triggered. And of course, post-marketing surveillance, because with any drug, with any vaccine, we always need to continue surveillance. We never treat enough people. So in some people, we might see some reaction. So this is why post-marketing immunosurveillance is very, very important. However, in terms of, in the case of COVID-19 pandemic vaccine has been accelerated. And this is something that often, if this pathway is taken often when there is a threat, and which is like COVID pandemic. So in this case, regulatory such as FDA do allow accelerated process. And this acceleration process involves combining the phases. For example, here, we don't need to submit a whole panoply of IND enabling study for FDA to allow us to trigger phase 1. This is as long as we show some toxicity data and efficacy in animal model, phase 1 safety is initiated and it's staggered with phase 2, which safety to induce and to generate safety as well as efficacy data. And before end of phase 2, phase 3 is initiated, And the approval process starts before even finishing phase 3. And this is, again, as I mentioned, this is due to the situation of COVID-19. And specifically in emergency use, also manufacture, instead of waiting approval, it started earlier because we need to make a lot of vaccine to face this pandemic. So this is why we didn't wait FDA approval to make the vaccine, but it started during the approval process. So the example of Moderna, Pfizer, BioNTech reached vaccine, they reached clinical trial within 63 days after selection of the sequence of the spike protein. And FDA granted emergency use in less than 1 year. Recently, the Pfizer vaccine, which is called Comirnaty, was granted full approval by FDA. So this is really never seen in lifetime of vaccine, to have a vaccine approved in less than one year and a half. And likely there was enough data for safety and also efficacy to trigger this approval. So even we have COVID vaccine, there is still many unanswered questions, because again, this is a critical situation that we had to come up with a vaccine, even though it still followed the required regulatory requirement, but there are still unanswered questions, which include first, the primary endpoint for mRNA vaccine. Sorry, I'm mainly focusing for mRNA vaccine since this is a new technology, but it is also applies for other vaccines that are currently developed or under development for COVID-19. So as I mentioned, the endpoint for vaccine trial was symptomatic illness. We just see that the patient is not sick because he got the vaccine. However, we don't know if this effectively prevents the transmission. We don't know if a subject that has been vaccinated, and even though he was in contact with the person who has the virus, he doesn't get sick, but we don't know if he has enough viral load to transmit the virus to another person. And also another unknown point is that the duration of vaccine immunity is not known. We know that immunity of COVID vaccine does persist for several months. However, there is emerging data indicating that immunity wanes over time. And there's many countries that are considering a third dose in order to keep the protection level high enough to provide efficacy. And also, as with any new drug or vaccine, long-term safety data will need years of vaccination follow-up to make sure that it's safe. And also, unfortunately, up to today, we don't have any efficacy data on safety in children younger than 5 years and in pregnant women. This is because right up to date, we have vaccines for 5 years and older. But 5 years and younger, we don't have vaccine yet, and pregnant women vaccine efficacy is still need to come. And also, current SARS-CoV-2 genome appear relatively stable, but we don't know how virus will respond to selection pressure of mass vaccination. Initial vaccine was produced against alpha variant. However, there was some additional variant that appeared, such as Delta variant. However, the mutation even though there was a mutation, the current vaccine was still protective because the mutation was not very big. It's still mutation in the targeted antigen, which is spike. However, current vaccine did protect it. Unfortunately, recently, there is an additional variant, which is Omicron, that has been identified with many number of mutation. And we don't yet know if the current vaccine is effective in preventing the disease. So currently, a lot of scientists worldwide are assessing this, and hopefully it's still preventive. If not, certainly there will be another vaccine to be made. And I think given the speed and what we learned from the first vaccines, we will be able to have a new vaccine shortly. And I believe the big pharma, such as Pfizer, the advance that they can have a new vaccine within less than 10 months. So, but let's be positive and hope that current vaccine is still protective against Omicron. So since the topic of my question is, if my talk is what's new from smallpox to COVID-19 vaccine. So I wanted to illustrate this with a picture. So the left picture shows it's taken in 1947. New Yorkers line up outside the Bronx hospital for mass immunization. And the right picture is people from Denver are lining in their car for mass COVID-19 vaccination. This was taken early this year, 2021. So overall, up to date, so Jenner's discovery of vaccination is still ranked as one of the greatest scientific achievements of all time. Vaccine is still the most effective way to prevent infectious disease and stop pathogen spread. More refined platform medium to safer vaccine and quicker to produce are currently, so this is the new, we're making safer and quicker and more efficient platform. However, from smallpox to COVID-19 vaccine, we are still dealing with technical and social adaptation. In Jenner's time, in our time, we have seen people against vaccine, and there are a lot of debate. And there was time in terms of smallpox where people have been forced to get the vaccine, and we are getting there with COVID-19. Right now, there's a lot of anti-vaccine people. However, they will certainly come to their sense because we can't do without vaccine. Otherwise, those little pathogens can make a big, can be devastating for humanity. So hopefully, this will be common sense will win over this. I think this was the last. Yeah, this was my last slide. So I won't be able to take questions since this tape presentation and it's not live. I will try to make the call on the day of the presentation. Otherwise, I'm willing to take questions by email and I can respond. Thank you very much.